So today I'm going to be talking about some of the really recent research I've been doing over the past 2-3 months on targeting the molecular motions of the BOAT1 ACE2S1 complex for rational drug design. So this work is related to COVID, and COVID right now it's a disease that needs little introduction. SARS-CoV-2 is the virus that is responsible for the current worldwide pandemic. It's caused over 10 million cases and 500,000 deaths worldwide over the past 6 months. There's been a worldwide scientific effort to try and understand this virus and create methods to treat and prevent the infections. My work has been focusing on how the virus enters our cells. So this is a representative model or figure showing the cell membrane of a human cell. There's a bunch of different receptors in this cell membrane, one of which is ACE2. The SARS-CoV-2 viral particle binds to ACE2, and this binding event is critical for the virus to be able to enter our cells. There's other proteins that mediate the entry of the SARS-CoV-2 viral particle into our cells. So the TMPRSS2 protease is one such protein, and this protein cleaves the spike protein of the SARS-CoV-2 viral particle to allow for the membrane of the viral particle to fuse with the membrane of the human cell in order for that virus to enter our cells. There's been a lot of research that has been looking at the binding interaction of the SARS-CoV-2 spike protein with the ACE2 peptidase domain. So the peptidase domain of ACE2 would just be kind of this blob area here. And so there's been lots of simulations and lots of crystal structures and cryo-EM structures come up about what is the interface between these two proteins, that spike protein and the ACE2 peptidase domain. However, this is just looking at it from one side of the picture. We're looking at the whole spike protein, but we don't have the whole ACE2 protein. So we can look at it from a different angle, and now we just have part of the spike protein, and now the entire ACE2 protein is shown here in this structure. So the peptidase domain would simply be this region here, but then we have this additional neck region as well as a transmembrane helix that is part of the ACE2 protein. There hasn't been any work looking at how, beyond this cryo-EM structure and the work I'm going to be talking about today, how the SARS-CoV-2 S1 binding domain binds to this whole ACE2 protein. So we're trying to look at whether the structure of this ACE2 protein, when we look at the entire thing, changes how that S1 receptor binding domain or the spike protein interacts with ACE2. But this ACE2 protein, we look at it within the membrane. There's this region that links the transmembrane helix, and the peptidase domain and neck region is this flexible linker. And that flexible linker doesn't look very stable when we just put in the membrane like this. And indeed, if we do simulations on it, the ACE2 protein collapses down onto the membrane. And so it's not able to maintain this structure. And this is because the ACE2 protein is not found in isolation. And the cryo-EM structure that has come out of the ACE2 protein actually has ACE2 in complexation with a membrane transporter, BOAT1. And the ACE2 BOAT1 dimer also dimerizes with another ACE2 BOAT1 dimer. And this is the complex in which, in the cryo-EM structure, they had seen the SARS-CoV-2 S1 binding domain bind to. One thing about this complex, though, however, is when this complex is formed, can no longer access ACE2 within the membrane, and therefore you can no longer get the TMPRSS2 protein to be able to interact with ACE2. And since the TMPRSS2 protein mediates that viral entry, we thought that maybe this dimer formation of the complex could prevent the viral particle entry into our human cells. And so we wanted to understand the dynamics of this complex and how each of the different proteins are working together in order to affect the SARS-CoV-2 particle binding to ACE2. So through our project, we were using molecular dynamic simulations to understand the structural determinants of the BOAT1 ACE2 complex and how these different factors modulate the viral particle binding and thus the viral particle entry into the cell. There's a few different cryo-EM structures that have been published of this ACE2-BOAT1 receptor complex. One of which is in the closed state, in which a substrate is bound to BOAT1, and cofactor, the zinc cofactor, is bound to ACE2. And this complex has the S1 viral protein bound to ACE2. However, when the ACE2 and BOAT1 were in the open state, so no cofactor bound and no substrate bound to BOAT1, the S1 protein, although it was present in the solution, did not bind to ACE2. So we wanted to understand if the substrate binding was influencing the ability for the viral particle to bind to ACE2 and how each of these different components were working together. So we did MD simulations on this receptor complex. So we put the BOAT1 ACE2 dimer and embedded it into a model membrane, and then solvated the system in water and 0.15 molar sodium chloride. This led to a complex that is about 800,000 atoms. It's absolutely massive. And then we simulated this complex using the Gromos 54A7 force field in GROMEX 2019 and ran 500 nanosecond simulations on each of our systems and ran each of those in triplicate. Using these simulations, we were able to look at how different factors affect this complex. So how the protein conformation, whether the protein was in that open or closed state, affects the overall dynamics of the structure, how the presence or absence of substrates affects the dynamics, the presence or absence of the zinc cofactor within ACE2, and also the presence or absence of the S1 viral particle. So I'm going to go through a few of these different factors and how we saw each of them affecting the dynamics of this complex. So starting with the protein conformation, the closed conformation is defined by having that substrate bound. So in BOAT1, we have a leucine substrate bound here shown in yellow. And when that leucine is bound, there's a network of hydrogen bonds that form within BOAT1 that prevent the leucine from exiting out to the extracellular solution. And this is a state along the pathway of the transport of leucine from the extracellular to intracellular solution. This contrasts the open conformation in which the leucine substrate isn't bound and that network of hydrogen bonds isn't formed, and this allows the leucine to be able to bind into BOAT1. The other difference between these structures is the binding of the zinc cofactor, where in the closed state, the zinc cofactor is bound and coordinated by some water molecules, histidine, and some glutamate. And so if we look at just the distance between the carbon centers of those glutamate residues, about 7 angstroms between them. But this distance is elongated in the open conformation to about 10.5 angstroms. And in that open conformation, we don't see the zinc being able to bind in that active site. So we've opened up the ACE2 active site here quite a bit. So those are the different factors of the ACE2 and BOAT1 individually that influence that open or closed state. We then looked at how this interacts with, influences the dimer formation. With the dimer formation, the only interactions between the BOAT1 ACE2 dimer and the other BOAT1 ACE2 dimer were interactions between an alpha helix and ACE2. If we look at a top-down view of this alpha helical interaction, within the closed state in blue, the two alpha helices are quite parallel and they interact with each other. But however, in the open state, we see a widening of the distance between these two alpha helices, that indicating the ACE2s aren't interacting quite as strongly with one another. We look at the hydrogen bonds that control this interaction between the two ACE2 monomers. Within the closed state, we have a network of hydrogen bonds along the entirety of the alpha helix. However, in the open state, we have quite a few hydrogen bonds down the one end, and as they get further apart, we lose a lot of the hydrogen bonds along those ACE2 alpha helices. We can look at how often those hydrogen bonds are formed over our simulation, when these are ordered from this end of the helix all the way up to this end of the helices, from left to right on this graph. You can see in that open state, all the hydrogen bonds that are occurred for more than half of our simulation time are all down that far end, so that's down here. Well, for that closed state, we are able to see hydrogen bonds along the entirety of the helix that stabilize that dimer formation. We then looked at the movements of each of these different regions through calculating RMSD of each region individually. And so the boat one, so this a low RMSD indicates low movement, high RMSD indicates we're getting a lot of movement in that region. So regardless of the open or closed state, the RMSD for the boat one subunits was about 4 angstroms. However, for the ACE2, we're seeing a lot more movement, and the RMSD is quite high in the open state, about 20 angstroms, but it was reduced in that closed state to about 13 angstroms. So having substrate bound seems to be stabilizing the dimer of dimers. So next we wanted to see how these differences in conformational flexibility and dimer formation affects the ability for the S1 particle to bind to ACE2. And so we took each of these, the open and closed state, and looked at it with the S1 receptor binding domain bound. So this was the crystal structure or the cryo-EM structure for the closed state, and for the open state, we manually docked that S1 receptor binding domain to ACE2. Over our simulations of these complexes in the closed state, see there's very little motion between, it's just vibrating slightly on the left. However, if you compare this to the open state, you can see that substantial conformational change occurring, particularly in the ACE2 domains. You can see the ACE2 domain on the left here is bending over quite a bit and moving towards that membrane. So we're seeing a lot more movement in that open state than what we see in the closed state. We can then look again at how the two ACE2 monomers are interacting. And again, looking at how those alpha helices interact in the middle. Without S1, so that's the data I presented previously, we saw that opening of this interaction in the open state. And this opening is observed again within that open state with the S1 particle bound. So the binding of S1 isn't really influencing either the substrate binding or the interactions between the two ACE2 monomers that I talked about previously that we observed without S1 binding. If we look more specifically at how the S1 binds to the complex, in our closed state, you can see that we have our zinc cofactor, we have our active site here, and then in red here, I've shown that S1 receptor binding domain bound. In the open state, you can see that opening of the active site within ACE2. And while we still have the S1 protein being able to bind to ACE2, it's binding in a different conformation. So the shape of this red surface is different than the shape of this red surface. If we look more closely at this, and we look at the hydrogen bonds formed along that interface, in the closed state, we get about 7 hydrogen bonds formed. We get 7 hydrogen bonds formed between the S1 and ACE2. And these hydrogen bonds are formed for about half of the simulation. In contrast, in the open conformation, only 2 hydrogen bonds are holding the S1 receptor binding domain to the ACE2 protein. And these hydrogen bonds are occupied for about 1/3 of our simulation. So there's less hydrogen bonds and they're less formed for less time of our simulation. So that S1 is not binding nearly as stably to the open conformation as it is able to bind to the closed state. We go on and look at how each of these different regions are moving. I have the open and closed state here without S1, so the data I presented previously. And then we can compare that to what RMSDs we get for the different regions upon adding S1. In all 4 systems, the boat one maintains the RMSD of about the 4 angstroms. So there's no change in the movement of boat one over the simulations. Adding S1, on the other hand, reduces the movement of the ACE2 domain for both the open and closed state. This is really evident within the closed state where ACE2 ends up having an RMSD of 7 angstroms compared to the 14 it originally had within that without S1. We also see a lot of movement of the S1 over our simulations. In the open state, S1 is not very happy to be bound to ACE2, and we have an RMSD of almost 30 angstroms, so it's moving an awful lot. However, in the closed state, it reduced by about half. So we're getting a 15 angstrom RMSD for that S1 particle. It's being able to bind a lot more stably to the complex. So what this is showing is that having a substrate bound to boat 1 really seems to be influencing how the viral particle can be able to bind to ACE2, seeing allosteric effects throughout the complex. One problem of this with looking at it with leucine is transported. So it won't stay bound within the boat 1 binding site for a very long time as it's transported through boat 1. And therefore, upon transport, we might get destabilization of the complex. And then that might allow for the S1 particle or coronavirus to be able to enter our cells. So we thought about what if we look at inhibitors of boat 1. So there's E4 is a competitive inhibitor of boat 1. And so it's not transported through boat 1. So maybe the binding of E4 into the boat 1 active site can have the same effects to stabilize the complex and prevent the breaking up of the dimer. So we're stabilizing the dimer of dimers without having the impact of it being transported through boat 1. E4 is an okay inhibitor of boat 1. It's really not that great though. Has an IC50, so the inhibitory concentration, which has 50% of its activity, of 7.7 micromolars. A good inhibitor we want in the nanomolar range. So we thought we'd try and understand how this is binding to boat 1 and if it has that same influence as what we saw with the leucine bound within that boat 1 active site. And then based on this, maybe we were able to rationally design a better inhibitor of boat 1 that could strongly inhibit the separation of this dimer. So if we look at how the leucine and E4 bind within the active site, in both cases, we have 2 hydrogen bonds that stabilize either the leucine or E4. The leucine there to the carboxylic acid moiety, and those same 2 amino acids form hydrogen bonds with the amide in E4. Leucine is also stabilized by coordination of an active site sodium ion. That sodium ion is not involved in the binding of E4 in the active site. And then in both cases, there's a bunch of aromatic residues that form non-covalent interactions and some stacking interactions with the leucine and E4. Well, the leucine is bound stably within one conformation in that active site. The E4 moves around quite a lot within the active site. And so 2 different conformations I've shown here in which we have similar stabilization of the amide within the active site. However, the aromatic ring moves from being in a parallel to a perpendicular orientation in that active site. So that's maybe something we can target in the future to try and stabilize and make a better inhibitor for boat 1. We then look at if the E4 is having that same effect on the RMSD of stabilizing the conformation. And we see a similar RMSD for boat 1 in the open as well as the closed with leucine and the closed with E4 states. RMSD about 4 for boat 1. And then the RMSD for ACE2 is similar in the leucine and E4 states where it is decreased by about 5 angstroms relative to that open state. And then look at if the same thing holds true when we bind the S1 receptor binding domain to ACE2. And again, when we E4 bound within boat 1 seems to have a similar effect to what the leucine bound within boat 1 has, with the RMSD of the boat 1 being about 4 angstroms, the RMSD of the ACE2 with S1 bound being about 8 angstroms, and that S1 RMSD being about 15. And these are all in both the leucine closed and the E4 closed state substantially lower than the RMSDs we were seeing in that open state. So in conclusion, the substrate binding to the boat 1 active site stabilizes the boat 1 ACE2 S1 complex, stabilizes dimer formation. And having this dimer formation is likely going to prevent the protease from being able to bind to ACE2 and therefore might prevent the viral particle from being able to enter the cell. And so this is opening up a new avenue that drugs that target boat 1 may be able to prevent the SARS-CoV-2 viral particle from entering our cell. And finally, I'd like to acknowledge the Omar group. So as our collaborators, as well as NCI for the special COVID grant to allow us to be able to do these simulations on this project, and all of you for your attention.
So I'll clap on behalf of everyone. Just multiply that by 69, because that's how many people we've got here.
Yay!
Yeah, thank you, Tak. Thank you everyone for all the awesome engagement. If you've got another question, please feel free to put it in the chat. In the meantime, I'm going to start on with the questions that we've got. Okay, so Miro asks, on slide 25, can you clarify how you are calculating the RMSD values? Is the reference structure the whole complex or just the protein of interest?
Yeah, so for these RMSD values, we have a line based on boat 1. And this that's the bit that's most in the membrane. So we did use that as a reference. So that is going to artificially lower the RMSDs of the boat 1 relative to the other proteins because we're aligning based on them. So the RMSD of ACE2 is also based on the structure being aligned to the boat 1 proteins.
And a related question that I noticed from a student, Jyoti's asking, how is this RMSD related to the ability of S1 to enter the cell?
So we're relating, so the RMSD tells us how much the complex is moving. And the hypothesis we have is if we have a dimer formation, then the S1 can't enter the cell because the other protein that required for entry can't bind. And so if it's moving around a lot, that means that maybe this dimer isn't stable. However, if we have a low RMSD, it might not be moving as, it's more stably bound as a dimer. And therefore, if it's dimer, we're not getting an entry of the virus into the cell.
And I guess that relates, sorry, I am structuring these even though they are going backwards. A question that I had is, do you want strong or weak bonds between SARS-2 and ACE2? Do you want weak bonds or strong ones?
So really the strong bonds are fine as long as we have that dimer formation. As long as it's not preventing the dimer formation, the S1 binding to ACE2, really either strong or weak. Okay. If it's weak, it's not binding at all. If it's strong, it's bound, but as long as we still have that dimer, the binding isn't going to lead to entry into the cell. So
weakness is bad. And there's a question from Peter, how often is the boat complex in an open state relative to a closed state?
So the open state is going to be when the substrate isn't bound. So within, it's going to depend on what it's transporting and the equilibrium within our cell. So the boat 1, it'll be in the open state, and then the leucine substrate or other amino acid substrates will bind, it'll go into a closed state, transport the substrate, and then it'll open up again in its transport cycle to allow the next leucine through. So if we're thinking of the inhibitors though, if we bind an inhibitor and it doesn't transport, then we might be able to lock it in the closed state and not have it open up again.
And a related one, do these pairs always dimerize or are they found singly?
So we don't really know. So this cryo-EM structure was, I believe, the first experimental evidence we had of the dimerization, but we don't have any structure of it not dimerizing. So we don't really have the evidence, I guess, either for how often either is formed.
And related, are water molecules involved in the hydrogen bonding between the helices? On slide 18 is the reference.
So I didn't see any water bridged hydrogen bonds. So yeah, that's those ones. So these are all direct amino acid to amino acid hydrogen bonds. There was no water mediating any of these hydrogen bonds.
And I think for our last question, you've got lots of questions this time, good work. Can you explain, from Martina Lesio, can you explain more about the membrane modeling? Are they actual atoms, coarse grain modeling, or are constraints to simulate the presence of the membrane?
Yeah, so we did united atom simulations. So we do actually have the lipids and cholesterol molecules explicitly represented in our membrane. We did a model membrane, so it's 80% POPC, 20% cholesterol. Other work we're doing on the system is putting it into a realistic membrane in coarse grain simulations to see how that's influencing the structures as well.
Excellent. So I might ask you to stop sharing your screen and let Chris share his screen, but I won't let you go quite yet because Adam has asked an awesome question to end on. What is the most exciting, fun, or thrilling thing to discover during the project?
So say for this project, this is an absolutely massive project, the biggest systems that I've ever done modeling on. And so to see it come together and see that impact that we're getting the allosteric effects throughout the protein and we're able to get this through modeling was really exciting.
Excellent. That's a really good answer. Okay, I might leave any other questions for afterwards. We can definitely leave the Zoom session on past 2 o'clock. I'm pleased to introduce Chris Hansen from UNSW, who's going to share his screen maybe. Well, he shared his screen. He might or might not share his camera. And he's going to, oh, you don't have to. And he's going to talk about an experimental topic.
I think my camera is shared.
Theory in there too. I'll leave him to talk. Excellent.
Okay, before you go, Laura, can you hear and or see me?
Oh, I can hear and see you. You're perfect.
Okay, excellent. Cool. All right. Well, good afternoon, everybody. Thanks for tuning in. And thanks, Tack and Laura, for giving me a chance to speak in the first episode of the webinar series. And so the first time I spoke to this community was actually in 2012 at a conference in Katoomba organized also by Laura McCamish. And so back then I was a student with Adam Trevitt. I've done a few things since then, and now I'm a lecturer and Decker Fellow at the University of New South Wales. So I'm also going to try and keep it topical, but I'll talk about the curve that we're not flattening, which is climate change. So you might remember in January last year, I spoke to you in Perth about some ideas I had for a Decker application looking at fluorinated compounds in the atmosphere. And that's just how long ago this was. I mean, that was me in a room with actual people. I mean, that was probably Jeff, that might have been Duncan. And I was actually able to cross the WA border and get to Perth and give you a talk in person. And so, I mean, thanks for that opportunity to do so. And the good news is that that application was successful. And so now I'll give you an update on some of the exciting early results we've been able to do from this project commencing at the start of this year. And so why are HFCs a problem? Why are they in the atmosphere? Well, until the early 90s, HFCs really weren't in the atmosphere. So they were proliferated rapidly as a solution to the ozone crisis. So CFCs, as you know, have been a refrigerant and fire suppressant since the late 19th century. And we worked out they were kind of, destroying the ozone layer. So we decided to flatten the curve. That ultimately finished in 1987 when we implemented the Montreal Protocol, mandating the global phase down of CFCs. And so what you notice here is the CFCs, they go away, they're banned now, and their replacement starts to go up. And the HCFCs, they're also ozone depleting, but they're a critical link to getting from CFCs to HFCs while the technology was developed. And so why don't I like HFCs? So they don't deplete the ozone layer, which is good, but they have very large global warming potentials. And so how do we quantify something's global warming potential? So we get CO2 and we set it to 1. So this is biomass. So 1 ton of CO2 emitted today contributes as much to surface heating as 1 ton of CO2 emitted today. If we go to a hydrofluorocarbon, so for example, something like fluoroform, which is also called HFC23, so what's its global warming potential going to be? And so I'm hoping Laura and I can work out how to use these Zoom polls. So I'd like to bring up the first poll now, if that was possible. Laura?
Yeah, no, I'm here.
Okay.
No, I want to do another one. Continue. No, continue. I'm getting there. End poll.
I saw it for a second.
Okay, well, you're just gonna get both questions at once because
okay, sure.
Right. Sorry.
But don't answer the second one.
Yeah, just answer the first one.
So bearing in mind CO2, which is set to 1, biomass, how many times worse is CHF3? So I don't know how we can see the results. Are they live or?
Well, we've got 3 people.
Cool.
4 people have voted. We've got to find the polling thing, everyone.
It's not, it just popped up for me.
Oh, okay.
Laura,
yep.
A lot of people are saying that they have to answer both questions before.
Okay, we'll answer both questions.
Take a guess.
Sorry, that's my stuff up.
That's all right. We're all learning. I guess when you go like a critical mass, we'll move on.
Yep, we've got 61%. Oh, good work. 70.
All right, I guess we'll pull out the results and move on.
Yep. Can I end poll?
Do we get to see the results?
Yep. Share.
Okay, cool. Look at that. All right. So I guess most of you realized it was larger than CO2 and by some large amount. So the quarter of you that said it was 12,600 were correct. So I can't progress my screen now. Why not?
Does that work?
I don't know. I'm trying to progress my screen now and PowerPoint's not working.
Oh, okay. I can't help you, PowerPoint.
Here we go. Boom. All right. So whoever said 12,600 was correct. So these things have these enormous global warming potentials. So 1 ton of this emitted today heats the surface of the Earth over 100 years as much as 12,500 tons of CO2. And so this is largely because they're very infrared active. It's in a part of the spectrum. CO2 is not very active, and they have these enormous lifetimes. And so this cylinder here, pictured, next to an average-sized American, holds 13 kilograms of gas. And so that's the value to society of this, is you can charge 3 air conditioners. If you were to empty this cylinder, you contribute as much to global warming as 10 Australians do in a year from their CO2 emissions. And so that's the worst HFC. Here's some typical ones. These are the ones expected to cause us the most grief over the next 30 years. And you see it's kind of interplay with how high their global warming potential is and how widely proliferated they are. These ones here, more modest GWPs, but they're in almost all air conditioners. If you have an aircon at home, you probably own a few kilograms of this. If you've got a car that's two or more years old, you probably own some of this fella as well. So these are really widely proliferated. But we're getting rid of them. So there's a phase down going on. So this is a complicated graph here. The main thing to focus on is this blue curve here, which plots the emissions of fluoroform as a function of year. So, it was rising up. We solved the ozone crisis, going up and up and up. So in 2006, the European Union banned these things in their F-gas directive, and it started going down again. But the rest of the world was still developing, population was going up, so it rebounded again. Until in 2017 or 2016, we amended the Montreal Protocol and made these things, mandated the phase down globally. You see, it started going down and then it just turned around. It's just increasing again. So this crosshatched area here is the reported reduction in emissions of these compounds. And as you can see, that the emissions just do not mirror the accounting that's done behind them. So if I'm going to cling to this metaphor of flattening the curve, this is kind of like a second wave of fluoroform emissions. And so, what else happened around here? So currently, this difference is being attributed to a particular nation not fulfilling its obligations. But what else happened here? Remember with the CFCs, they went down and something else came up. So the replacement this time, which you might not know about, is the hydrofluoroolefins. So these things are huge. And so what they do, they incorporate a double bond, which makes them more reactive, decreases their lifetime. And so they don't get to the upper atmosphere and they don't, contribute to surface heating. These two here, they're both propene derivatives. They're the replacements for the one that's in your car you see at the moment. So no ozone depletion potential, no global warming potential. They react away in less than 2 weeks. They don't get the upper atmosphere. So you give these numbers to your regulators, and the regulators approve the compounds, and they start becoming very industrially important very fast. The first of which is called HFO1234YF. It's just this fella here. So it's flammable and it burns to yield HF. So this is a problem because it's designed for automotive applications. So if you crash your car and the refrigerant catches on fire and makes a toxic gas that rots your bones from the inside out, this is not an industrially acceptable property. So they blend it with these blacklisted HFCs, which is kind of not solving the problem either. It also reacts in the atmosphere to make trifluoroacetic acid and 100% molar yield. This is persistent, it doesn't biodegrade, it's toxic to marine life, it's harmful to humans, but still we're accepting it's not so bad. So almost all new cars since 2018 use this as their air conditioning refrigerant. We are developing a replacement, which is it's been available for about 10 years now, called HFO1234ZE. So that's an isomer, it's also a fluorinated propane, also got a 0 GWP. So it's not flammable, which is great. It doesn't make trifluoroacetic acid. And so it's becoming very important. So our own Department of Agriculture has recommended it for foam blowing for the last 7 years. It reacts in the atmosphere to make trifluoroacitaldehyde in 100% yield. And this CF3CHO is removed from the atmosphere 4 times faster than its progenitor by photolysis. So what do we actually know about the photochemistry of trifluoroacitaldehyde? Well, the studies going back to the 1950s confirm at 308 nanometers, which is just a historically simple to generate laser wavelength, you have access to this radical channel making trifluoromethyl and formal radicals. Some studies observe a minor molecular channel making fluoroform and CO, but most 21st century studies dismiss these results. This would be an absolute disaster. So 100% of these refrigerants make trifluoroacitaldehyde, and if a small amount made fluoroform, the GWP is going to be significantly increased because this is almost 13,000 times worse than CO2. So it's important to mention that these previous studies are indirect, so they're done offline. They put a radical scavenger in, they try and scavenge the radicals and look for the radical adduct in, say, FJR, for example. And so this demands further study. This is just a reminder that this thing's so persistent, it's purely man-made and we can see it in space. So carbonyl photochemistry. So the current understanding of trifluoroacitaldehyde's photochemistry is simple. When we teach undergraduates about complex photochemistry, usually is the example of the carbonyls. So you remember the Norris reactions and whatnot. If we look at the non-fluorinated analog, acetaldehyde, it's got a very rich UV photochemistry, and this just demands further investigation. So I guess you've already answered the question now, but you remember last year that Scott Cable forced us all to remember precisely what the high energy of the highest photon that reaches the Earth's surface was. And so I'm guessing people guessed. I don't know if you can bring the poll up again, Laura. Is that easy to do? Like the results?
Yes, actually.
See how well people remember.
There we go. Can you see?
Yes. Okay. So
I didn't look it up, so I don't know the
yeah,
answer.
okay. So I think if we asked the question last year, I think almost everybody would have guessed 400 kilojoules per mole. So I guess I threw everyone with the trickier specific wavelength. Anyway, so yeah, so
You threw me.
that's just going to work again. Right. So if you said 400, you were right, and 1 in 4 of you did. So let's explore the potential energy scheme for this molecule. I don't want to say surface after the last talk. And so I've included the upper wavelength limit, or the upper energy limit, as well as the wavelength that all the previous studies guided and regulators have used. And we'll start putting some stuff on here. So the dominant channel is the radical channel making, methyl and formal. This actually proceeds via a triplet, on the triplet surface via a barrier. And so do we see a similar kind of landscape for the fluorinated species? Yes, we do. It looks almost identical, bearing in mind that the acetaldehyde is experimental and the fluorinated one is calculated. And then once you go below this triplet barrier, you turn on a much richer ground state photochemistry. And so some of the channels you see, so there's a transition state leading to methane and CO, which is analogous to the fluoriform channel that might exist in trifluoroacetaldehyde. Yeah, and you see it looks similar again for the fluorinated species. You might lose a H atom, similar energetics once again. There is a barrier leading to a ketene and molecular hydrogen. I couldn't find this one for the fluorinated species, but there was a different pathway leading to, it was largely the same, but we're just missing a carbon-carbon bond to make, the triplet difluoromethylene, CO, and HF. So qualitatively, the energetics appear quite similar for these two species. And so then, what next? And so in acetaldehyde, is this going to work? Don't play the video. I do want to play the video. The transition state pathway is available, but it actually proceeds through 2 roaming channels. And so what roaming is, it's kind of like a frustrated radical reaction, where you break a bond, but then the photoproduct can't escape the kind of long-range weak forces of the cofragment, and it comes around and kind of does like an intramolecular reaction to form photoproducts. I wonder if I can play that again. Cursor. Screw it. All right. And you can see here we have this internally active now ground state molecule. It's it's undergoing vibrational redistribution. Eventually, you get enough energy to break this bond. H atom kind of roams around. It can't quite escape. Then it comes and snaffles this methyl group from the radical. You make this kind of internally active methane, and they drift apart. And so there is also a methyl roaming channel. I won't show the video. And so these are the 2 main pathways making methane and CO in acetaldehyde. So now we should test the same photochemistry in trifluoroacetaldehyde. So there's so many things we could do, but the important question to ask first is fluoroform actually a primary photoproduct? And I mean, all the data guiding the regulator so far is at 308 nanometers, even though that's, above the barrier at which we turn off, most of the, or decrease the importance of the exciting rich ground state chemistry. We should do that first. Having the radical products in a bulb adds uncertainty. They could react with one another. They could absorb another photon. So we should do isolated molecules in a molecular beam, which means we need to ionize the photoproducts to manipulate and detect them, but we can't ionize fluoroform because it's not a stable cation. If we had a technique where we could measure the internal and kinetic energy of the CO co-product, then we can infer everything else from conservation laws. So we'll use VMI. So that stands for velocity mapped ion imaging. So you can imagine a time of flight mass spectrometer with a position sensitive detector. So we have a jet cooled molecular beam, which we get into our vacuum chamber. We intersect it with a couple of lasers, the first of which initiates some sort of photochemistry that makes new photoproducts. The second or third or whatever, the next lasers come in here and they make ions. And typically they ionize specifically a quantum state of a certain radical or molecular species. We then accelerate them towards our time of flight region, where they're mass separated, and they impinge upon this detector. We configure our electrodes so that ions hit the detector according to the velocity that they're born with. So fast ions hit the perimeter, slow ions hit the center. So we get their mass and the time of flight, we get their velocity, which gives their kinetic energy. We pick a quantum state, so know their internal energy, we know the energy, the photon energy, the laser. So then we can work out the kinetic energy, the cofragment from conservation laws, and the last thing left is the internal energy, the cofragment. So if we can do this to CO, we can see if we're making fluoroform. This has been done for, so what do the data look like? This is one example looking at the photodissociation of H2S at 130 nanometers, detecting singlet B sulfur. So you get these rings, and so you integrate this radially to make a translational energy distribution for the sulfur atoms. And so each of these rings corresponds to a peak in the distribution, each of which corresponds to an internal energy state of the cofragment. And so I mean, it's a pretty gnarly example, but it's just fun, and you can kind of see, put the combs for the different, vibrational and rotational energy levels on here. You can see they kind of line up with the peaks in the energy distribution of the sulfur. So this technique's being used for acetaldehyde, but imagine it in a two-dimensional way, where rather than doing it at one photolysis one detection wavelength, we do it at several. And so you've kind of rolled the entire velocity distribution into a straight line now. So this would be your speed axis. This would correspond to slow things. This correspond to fast CO. And by changing the wavelength, we can ionize a different quantum state of the CO. So this is a rotational number here. You see two distributions. You see, kind of down here, we have slow stuff with a very low rotational quantum number. Then we also have stuff here with a broader distribution of velocities and with much higher quantum numbers, a higher rotational quantum number. And so what this is, hopefully everyone's following. So we can slice in a few ways. If we look at just the fast stuff, then we get our different rotational quantum numbers for CO listed here. And you see they're higher than 10. We slice it here and look at the slow stuff, we see we get this narrow cold distribution. Likewise, we can look at the different speed components for each quantum state range. We can extract so much information from this. And so we can do the same experiment for the trifluoro. But what I should mention here is these two different distributions. So this slow cold one has been worked out to correspond to H atom roaming. And this much more broad velocity distribution, hotter CO, has been worked out to come from methyl roaming. So we'll go to the lab. So normally when I synthesize stuff, I just buy it from Sigma, but you can't buy this one. You can buy the hydrate, which is not as easy as it sounds because it's actually a very stable diol. But you can put a bunch of concentrated sulfuric acid, P2F5. You can crack the hydrate into the gas you want. You can trap a few different things at different temperatures. And we did actually manage to make this thing and verify its purity by FTIR. So now we can go and do these 2D REMPI experiments, as they're called. And so this is the same experiment done on the trifluoroacetaldehyde. And you can see qualitatively it looks very similar. You have a slow cold distribution of CO photoproducts. But then you might notice that the methyl contribution seems less important in this case. We can integrate and get a spectrum, which is proportional to the quantum state distribution. You can see indeed we have less stuff at a higher CO rotational quantum state. And then we can also integrate the different speed components to form this here. And you see qualitatively it looks very similar to that for acetaldehyde. So this is starting to suggest that you might be making fluoroform for the same channels that you make methane and acetaldehyde. So this is a very important result, so we need to be sure. And it looks a lot like acetaldehyde. And so we used acetaldehyde to tune up the instrument. And as Nana used to say, if it looks like acetaldehyde and swims like acetaldehyde and quacks like acetaldehyde, it might actually be leftover acetaldehyde. And so one way we can check this is so we're one of the few labs in the world that can make these very heavy photons. So we can make an almost 11-volt photon at 115 nanometers with some custom equipment we have, and we can ionize all quantum states of both radical products, CF3 and CHO. So we can measure their velocities, we know their mass, we can get their momentum, and we know that the momenta have to be the same, that's the law. So what we do is we record an ion image for all quantum states of the formal radical, all quantum states of the trifluoromethyl radical. We can plot their momentum distributions and they agree perfectly. And so I've got some text here, but this basically shows us that the formal are recoiling from the trifluoromethyl. And then this is the only reaction in town. So if this is recoiling from, CH3, which is 4.5 times lighter, it would take a greater share of the momentum, just because the momentum being MV. We can also infer that, the CO we're seeing is not from hot formal dissociating because for the same reason, the H atom will take most of the kinetic energy. And so the CO distribution would look like this ring you see here. So now we're pretty sure that we're seeing fluoroform as a primary photoproduct of trifluoroacetaldehyde photolysis. There's less methyl roaming, so that's fine. We can also investigate the dynamics. I think we've shown we want to, but this is interesting. And so you remember that unusual triple fragmentation pathway I mentioned earlier, making the methylene, fluorinated methylene, CO and HF. Well, if you look at that transition state, it starts off with an elongation of this CC bond, like how you would start methyl roaming. You see, you start to form like a hydrogen bond between the CF3 group. And they leave us these three species. So while you watch that, I'm just gonna have a drink of water. So I mean, this is cool. This explains why you might be slowing down or, limiting the formation of the methyl rowing product, because the methyl can't quite escape because it gets tied up here. This is troubling because it also makes CO. CF2 and HF are also challenging to ionize. So I mean, this is unlikely to happen. It's entropically quite unfavorable. There's a highly dimensional surface for these things to move around on. The chance that they'll find this tight transition state is low. But what else can we do? We need to verify that we're not seeing CO this way rather than just wave our hands at it. So we can use FTIR. So we have an FTIR spectrometer here with a hole in it that we have coupled to a tunable laser system, so we can kind of in situ photolysis and then look at the products in FTIR. So this is not a good way to measure primary photochemistry. So for example, fluoroform could arise from, say, a CF3 reacting with the parent. So they're all in here. This is kind of like a more 21st century bulb experiment. However, HF is highly infrared active. It might be a product of some radical chemistry I can't picture, but it's unlikely to be consumed by it. So if we don't see HF, then we don't make HF by either molecular or a radical channel. And if we do, we got to think. So this here's a difference, infrared spectrum. So basically you've subtracted before photolysis from after photolysis, and you can see, the parent peaks go down. There's a few photo products like CO, CO2, CF2O, some other fluorinated species, but it's kind of meaningless what these are because the whole soup's in there getting hit with a laser all the time. So this is not really representative of primary photochemistry. And so, but the important thing is out here where you'd expect to see HF, you don't see anything, and it's just a literature spectrum of HF. You see it's fairly intense, about twice as intense as CO, and we don't see anything. So still unconvinced. What can we do? Well, we can make the reaction a whole lot more complicated by just putting a lot of oxygen in there. It's going to do something. It's going to change the products. There's much more CO2, sorry. We do see a little bit evidence of HF. And so if we zoom in, overlay the spectrum, so we are sensitive to HF. If we were making HF, then we would see it. And so then currently my discussion topic for you now is prove me wrong. And so I guess Laura tried to get me to encourage discussion. So if you want to, in the chat, try and convince me otherwise, that'd be fun, maybe useful. But now let's just wrap this up quickly, I guess, because I must have talked too long. Right, so like in most chemical dynamics, we're very sensitive to the one thing we're looking for. So we can characterize a reaction exquisitely, but we have no idea how important it is relative to another one. But, we can compare, these behave similarly, so we can compare them. So inspired by my barbecue bottle, we have the swap and go gas for chemical dynamics. So basically what we do is we put CF3O in, look at some particular quantum state of CO, turn our nozzle on and off to try and get, kind of a measurement for the intensity of that CO signal. We then run helium through it until everything goes to nothing. Then run acid aldehyde, and so we kind of get intensity for the CO from acid aldehyde. Assuming nothing changed, and you can get an idea of the time scale. We then kind of now work out a quantum yield. How do we do that? Well, we get a known quantum yield from literature. We get the ratio of our intensities, the ratio of the known, absorption cross sections for these molecules. We work out a ratio of how much the particular quantum state we're looking at compares to the total quantum state distribution. And so we can map that out, and it turns out the quantum yield for CO formation at 308 nanometers is 3.4%, plus or minus some error. Bearing in mind, this is aggressively not a quantitative technique. So what's the consequences of this? Well, they must be profound. So in the EU, refrigerants with a GWP more than 150 are banned. And this sets a limit in an upper limit of 500, but a likely effective global warming potential in the hundreds. So, and this also might account for the currently unidentified source of fluoroform in the atmosphere. So these results are profound, and we're working on it still. So we're going to go to longer wavelengths and do the same experiments. And so the analogies of acid aldehyde have held strong so far, and they suggest the CO quantum yield will increase, or fluoroform quantum yield will increase at these wavelengths. We want to explore some reactions with atmospheric compounds. We're also discussing with some industrial and academic researchers on how to communicate and verify these results. And I guess I was going to say, if anyone's interested in this, maybe you can email me, you can discuss scholarships we can look into, or projects. So move on from there to conclude, because it's almost 2. Obviously, a lot of people have helped, everyone in this photograph and many more. So that the main people are, Geody Campbell's a PhD student with Scott, has done most of the work here. Obviously, Scott and Tim, mentorship has been enabling. Laurie helped with the FTR experiments, and everyone else helped, and many people I've forgotten, including colleagues elsewhere in UNSW and the workshop and the ARC for funding it. So I guess that's all I wanted to talk about. Thanks for listening.
Thank you very much. I'll do the clapping for everyone again. Okay, so I think actually we had a really great example here of Jyoti, who you've just acknowledged, was actually in the chat and able to help out with some of the questions. So
Oh, thanks, Jyoti. I had the chat closed.
yeah, no, I think it was really good. Okay, I'm just going through the chat and trying to figure out the good questions. What's the concentrate that Jyoti hasn't completely answered? What's the concentration of CHF3 in the air currently?
CHF3 in the air currently. Yeah, I don't have a number for that actually. We could probably go and work out the emissions from that previous slide, but to be short, I don't have an answer for that.
Okay.
Yeah.
There was some questions about a question that wasn't answered. How are the global warming potentials calculated? Was it theory, experiment, combination of both?
Well, it's a combination of experiments and modeling. So they look at things like the reaction rates with, say, OH and whatnot. They look at the photolysis rates, things like that, and they can feed these into models to work out a lifetime.
So they're figuring out the lifetime. What about how much?
Oh, they can look at the absorption and emission infrared cross sections for the molecules, and they can model it. I think we're pretty good at this.
Yep. Oh, we have a list on the Google Sheet. Okay, good. Excellent. Okay. How do you control the timing between two lasers in a VMI experiment?
Okay, so these lasers are about, the pulse duration of the laser pulse is about 5 to 10 nanoseconds, which is well within the limitations of like timing electronics. So we just have a device with a very fast internal clock, and we can just choose the delay, and it sends a trigger to the laser and tells it to emit.
Cool. And Meredith asks, can you rationalize less roaming in CF3CHO?
Okay, I tried. Was that before my slide?
Yeah, it was probably before your slide.
Okay, yeah. So my thinking is, if this will play again, is as this bond's elongating.
Oh, it was, sorry. Meredith says you've answered it. Sorry, I was a little bit trying to keep everything together. It sometimes means I miss the answers. Okay, so I might leave it there and invite everyone. Chris, if you can stop sharing your screen, and I'll just share this one. Oh, goodness, how do I share it? Google Chrome, share. We'll just share this bit. I don't know how to present it very easily. Can everyone read that? Basically, Sarah Masters is going to talk to us next week, next fortnight, in one fortnight, on utilizing the combined power of theory and experiment to understand the quirks of molecular structure. And Peter Krause is going to talk about I Psi with my little I, about Psi version 1.4. Thank you very much, everyone, for the engagement and for coming along. This was a really huge turnout. Thank you very much for that.